We'll go ahead and move on to our last panel discussion. Thank you again to all the speakers of this session. Those were wonderful presentations, and looking forward to hearing your thoughts on some of these questions that Jeremy will be proposing, as he is our next moderator. I will turn it over to you, Jeremy.
Great, thank you very much, Emily. Yeah, so thank you again to the speakers in the last session. That was really interesting, and obviously a little bit different, where we were kind of moving and focusing our discussion on somewhat chronic questions as well. I think let's just start the discussion off thinking about chronic immune responses. Maike, I'm going to go to you first. I mean, one of the sort of, I guess, focus there is particularly with these chronic diseases like HCV, HBV. The DAA approach, seems to be working. It's a kind of, as I understand it, I'm not an expert virologist, but as I understand it, it's a complex mixture of different antiviral drugs that obviously are relatively effective in being able to suppress that chronic infection. But is that something that's going to be appropriate for all types of chronic viral infections, or is that going to be very much a what should our strategies be to kind of look at these different chronic diseases, chronic viral diseases?
So I think every chronic infection has its own peculiarities. So I think there is not one strategy to fight them all. I mean, for HCV, I mean, we are really lucky to have these DAAs in hand, and it's normally a combinatorial treatment and just antiviral. In terms of HPV, that's much more complicated because the virus can kind of hide from so because it can integrate, and then it's the episomal CCC DNA form. So we have the nature of the virus is really determining what we should have for strategies. And I mean, at the moment, so the most promising strategy seems to be to combine antivirals with immunotherapy, so that you have like really these two arms. And of course, there are different approaches. So for the immunotherapy, they are stimulating the innate immune system, or therapeutic vaccination. So there are different strategies, and that are currently under investigation. But the principle of combining both seems to be the way to go, at least at the moment.
Yeah, no, that's a very good point, actually. So in your perspective, then, what type of immune therapy might, like TCR specific, is that too patient specific? Are you thinking more biologics, or just wondering what kind of immunotherapy you think is going to be particularly useful?
Oh, I think so. I do not think that you really that it's So as you mentioned, if we really do like T-cell transfers with specific TCRs, that it's really like too personalized, I would say. So
very
we have
expensive.
to go, very, very expensive, not really feasible. So I think we should go broader. I mean, there are some promising results concerning therapeutic vaccination, but I mean, we have to see. And also in the context of HPV, what I mentioned before, like to trigger innate responses, since HPV is known as a stealth virus, so it does not induce that much innate immune responses. And therefore, if we maybe release that break, and then can induce the adaptive arm. But again, I think what Daniela mentioned in her talk with COVID, that the orchestration and really the coordinated immune response is also important in the chronic setting.
Yeah, it's very interesting what you were saying there about the importance, obviously, of the innate immune response. That kind of ties in very much with what Akshay was just saying. And from a lot of the work that we're doing, I mean, a lot of the discussion today has obviously been focused on desired immune responses and vaccine responses or against viral infection. But of course, in the context of unwanted immune responses against biotherapeutics, there's a huge focus on the delivery of relatively inexpensive generic peptide-based drugs. And regulatory authorities are very, very interested in understanding the potential impact of any impurities in these peptide products which can trigger innate immune responses, particularly in relation to dendritic cells, because that triggering can then lead to obviously the whole cascade of T cell responses, which then lead to antibody responses and will neutralize the effects of those therapeutics. So it's just another balance of the whole thing, and often the two sides of the party don't often mix. So, you know, today we're sort of very much thinking about wanted immune responses, but a lot of what we're thinking about all the time is also thinking on the other side of the equation. So it's very interesting to hear about those kinds of strategies to induce innate immune responses for vaccination as well, because it absolutely makes sense and is probably an area which has been quite significantly neglected. So thank you for your comments. Linda, can I come to you? So I think it's really interesting linking that leukemic patient and your case study there, and thinking about the really fascinating situation of a year-long infection of COVID. I mean, that's pretty interesting, but poor patient. And you mentioned it at the end about how that potentially link, you know, what can we learn from that specific situation that could potentially be applied in other areas, because obviously that's just one case study and everybody is going to be slightly different. But it seems to me that understanding these situations alongside one another is really important to help our gain understanding, rather than to have a very reductionist approach of like one disease at a time. What do you think a bit more about? Could you maybe expand on that a bit more?
Yeah, I mean, I think these, you know, different scenarios, obviously they do give some insight into the importance of the different arms of the immune system, but obviously that is a single patient, and you know, it did very much seem that sort of interesting combination of having no antibodies but having a T cell response, you know, might have played a role in the way that, you know, in the disease course that we saw in that patient. But then equally, you know, of course, we didn't look at innate immunity. I mean, that's something that as an adaptive immunologist, we often kind of like forget that there's a lot more going on. And you know, of course, the patient, you know, had other pre-existing diseases and had previously been very, very ill as well. But yeah, I think it does give us some glimpse into kind of you know, the importance of different parts of the immune system. But I think, you know, actually picking up on what Daniela said earlier, I don't think it's about kind of which is the most important. I think there is that sort of coordination going on between different parts of the immune system, and actually, you know, when one part is not functioning so well, that actually, you know, there is this amazing level of redundancy, isn't there, within the immune system, where another part can kind of help out a bit more. So I'm not sure that these kind of examples provide all the answers, but I think, yeah, it gives us some sort of glimpse into what's happening.
Thank you. Yeah, that's great. Akshay, what again for you? What do you think in the context of? I think particularly some of the challenges with the different coronavirus variants, the variants of concerns that we've been talking about as well. How, from your perspective as well, in the work that you're doing, how does that kind of translate into the effects of what you're looking at?
So in the context of,
sorry, Linda. Yeah, sorry. The question is for Akshay. Sorry.
Sorry. Apologies.
No, that's fine. It was my fault for not being clear enough.
So, Dr. Fry, the thing is, as we all know, that the T cell responses are largely conserved when it comes to the cross-reactivity with the different variants. There has been numerous papers which have shown that the cross-reactivity still remains conserved even in the even from the epitope point of view, that most of the epitopes remain conserved. So now the approach should be to look at those immunodominant epitopes and to look at those important regions which remain conserved across pan-beta coronaviruses, and they should be targeted again and again, so that the variants, anyway, it's quite likely that individuals who have been exposed to the virus will not have a severe infection because the T cells usually survive for a very long time. It has been observed that those individuals who have had a SARS-CoV-1, they had T cell responses against those even 17 years after the infection. So it's quite likely that at least in a small proportions, the T cells will still survive. But now the idea should be to because the space inside anyone in his anybody, the T cell space is limited. So it is quite probable that it might be eliminated. So we should be the approach should be that the T cell specific stimulations should be provided regularly in at least 2 to 3 years, so that the coronavirus problem should not occur again, at least in our lifetime.
But is your point that you're thinking of wanting to have just specifically a T cell epitope vaccine that's going to be appropriate for all different variants? Because then if you did have that, then you wouldn't necessarily be inducing appropriate B cell responses as well.
I agree to that part, but eventually we all have at least been vaccinated a couple of times. So the next approach should be, since the B cell responses are going to be diminished after some time, the antibodies go down. Now, if we are looking out for the individuals, such as for those individuals who have been not, they'll not have severe infection, at least they'll be infected. It might not be a sterilizing immunity, but those individuals will be mildly infected. So a T cell based approach would be better in this particular scenario. Obviously, some compartments of B cells or the conserved side of the RBD domain, which has been conserved in all MERS, SARS-CoV-1 and SARS-CoV-2, can be used to stitch them together and make a pan-beta coronavirus vaccine. But eventually, I am a little skeptic in that scenario that it's not going to be effective because virus is always going to learn and mutate and skip those particular regions. And as even as shown by Linda, that the T cell epitopes are not being pressurized and the mutations are not happening in the T cell region epitopes. It's not driving mutations in that scenario. Is quite interesting that that thing is at least not happening. So we can at least stick to T cells, keep that T cells at least in priority for a vaccine, which can be pan-beta.
Yeah, that's interesting. I mean, one of the things that I was wondering what everybody thinks about is that when we look at specific T cell epitopes that have been described for spike and various different open reading frames of the virus, it's very clear that there are very, very robust T cell responses restricted to certain HLA types, and then others, such as our friend HLA AA201, are much, much less. So if we look at some of the immunodominant epitopes for A2, they're not actually that strong. So not all, but pretty much most of the data we've looked at today has been based on overlapping peptide pools, looking at total gamma production, and that's fine. You know, you're looking at all the peptide presentation on either class 1 or class 2. But when we're looking at specific T cell epitopes, be it class 1 or class 2 restricted, and if particularly we think about CD8 restricted responses, it's quite clear that there are certain alleles which give really, really strong T cell responses and others which aren't. And if you happen to be an individual such as A2 positive and you don't see a really, really strong T cell response, maybe that's where the balance of the antibody response is more important. We don't really know, as far as I'm aware. I mean, I'm not following the literature super closely, but I think that combination of HLA alleles is probably quite important. Linda, you're very much into that thinking as well. I'm just wondering what your thoughts are for that.
Yeah, actually, I probably don't have a lot to add to that. I mean, I don't know if there's been any studies yet looking at kind of correlations with disease outcome and HLA type. I guess that's the sort of thing that we need. I mean, there's studies like that in, you know, diseases like HIV. Obviously, there's good correlation, but then that's a chronic infection, so that's probably very different. But yeah, it is interesting what you say about A2. I mean, we've been trying to look for good A2 epitopes and, you know, struggling really to see responses. And I think earlier on, I can't remember what talk it was in, but someone was showing staining with A2, YLQ.
YLQ, yeah.
And you do see some, but it's very low frequency. But interestingly, then when we grow, so we can have samples where we literally see almost no tetramer staining, and then we culture them for kind of 5-7 days, and then the tetramer positive population is massive. It's like 20% or something. So they are there, and they respond really well and grow really well. But yeah, I'm not really sure what that means.
It's interesting. And certainly, I think some of the very early papers from Tao Dong and Yongchung Peng's group in Oxford, when they were looking at pentamer staining and B7, B40, those epitopes there, the ex vivo staining is super, super strong, and A2 is very, very weak. And that certainly is something, like you say, you can expand those cells out. But yeah, it's an interesting phenomenon, which I think we don't really understand what the impact of that is yet. So
maybe we just haven't found a good A2 epitope yet.
Maybe there's something lurking under the scene. Yeah, but, yeah. Or maybe it's just that A2 is not the allele to be focused on. Unfortunately, it's the one we're interested in. Maike, what about you? Do you have any thoughts on that?
So actually, we did some epitope specific analysis on SARS-CoV-2, but we used like the same approach that we did for the chronic infections by tetramer staining and then enrichment with magnetic beads, and then we received some decent populations to do then further analysis. And there is an interesting thing that you mentioned with Tao's paper with this B7 epitope, that this is exactly a cross-reactive epitope with the common cold coronaviruses. And when we did like ex vivo frequency calculations, we can really see that the frequencies of T cells targeting this epitope are higher compared to the others. And we had epitopes A2, A3, A1, B7 and B35 restricted. And what we also see is that they are low in frequencies, but they are there. And they also differ in their phenotypes. If we look at HLA-B restricted T cells compared to HLA-A restricted T cells. So I completely agree with you what you said, that the combination of the HLA you have may impact then your response. I think that I remember one study that tried to do like an HLA association study, but the problem is that you have so many co-founding factors that it's hard really to then get the association, and therefore you can better see it in like subgroups, but then you do not get, of course, these associations. But,
great, thank you, Maike. I just want to bring it. Stephen's got a question, which we'll let him ask.
Oh yeah, thanks very much, Jeremy. Yes, but I had a question for Maike. You very sort of tantalizingly gave us a glimpse of CD74. Can you tell us anything about CD74 in your work?
CD74. So I can give you. I haven't shown data about CD74, CD127. CD127, but CD74, it's quite interesting because it's an interference stimulated gene, so it's really now just by chance that I can tell you something about that. And we found it upregulated in terminally exhausted cells, but we do not know at the moment what this means. But yeah,
you've struck gold there, Stephen. You've asked exactly the right question. I don't know how you.
Yeah, really.
I could have sworn I saw it in one of your figures, so that's what.
Maybe you saw it in the signatures.
Yeah,
and yeah, this is exactly why we are interested in this. And there are some ISG actually upregulated in these terminally exhausted, but it's not that we only have like this interferon signature in these terminally exhausted cells. It's also in this memory-like cells, but it's different sets. And yeah, it has something to do that they may also have some different roles besides antiviral activity. But yeah, we do not know exactly.
While I'm still on, could I also ask you, Maike, about, is there any similarity with CMV infection, chronic CMV infection at all that you know of?
Yeah, so no, it's really different. So it's at least in terms of this the signature and the phenotype, because you have like this latent activation in CMV with this memory inflation, and you have like really this persistent stimulation, and this obviously drives different programs in the T cells.
Great.
Right, okay.
Well, thank you very much, Stephen, for your question. I'm very conscious of the time, so I'm just going to first of all thank our panelists for joining us for the discussion, and for all the speakers for their time today. Actually, you're a star legend here, staying up. It must be goodness knows what time with you, so it's time for bed. Thank you so much for everybody who's joined us today. We really appreciate it. The talks have been recorded, so they will be available via our website and on YouTube in the next week or so. So thank you very much for joining us. If you want to stay in touch with people, a good way of connecting, we have a Mastering Immunogenicity LinkedIn group, so do join that, and then you can kind of connect with like-minded people and people who are thinking in slightly different ways as well, hopefully. But we really appreciate you joining us today, and thank you very much. We hope it's been productive and that you've learned a lot as well. Certainly I have. So thank you very much indeed for coming.
Thank you.
you.
Bye-bye.
Thanks,
Thank you.
everybody.
Thank you. Thank you.
Bye.